This recording on gene function will focus on the process of translation. Translation is the process in which the cell synthesizes proteins following the transcribed copy of the gene, the messenger RNA. Translation takes place in the ribosomes. Remember that in eukaryotic cells, ribosomes are found in two places, in the cytoplasm of the cell and attached to the endoplasmic reticulum, making rough endoplasmic reticulum, or RER. On the other hand, in prokaryotes, ribosomes are also found in two places, in the cytoplasm and attached to the cell membrane. Translation is the process by which the sequence of nucleotide bases, A's, G's, C's, and T's, are read as a code that will tell the cell the amino acid sequence of a particular protein. The cell reads 3 nucleotides at a time. These 3 nucleotides are called a codon. Each codon represents one of the 20 amino acids. The correlation between a codon and an amino acid is what we call the genetic code. So, A's, G's, C's, and T's, organized into sets of 3, which we call a codon, represent to the cell one of the 20 amino acids. This is the genetic code. In the genetic code, you have all of the possible combinations of A's, C's, G's, and U's. The genetic code is written in RNA language because it is the messenger RNA that will be translated, not the DNA gene. The genetic code is thus written in RNA language. Instead of thymines, the genetic code uses uracil. These are all of the possible 64 combinations that we can have of the 4 letters. We have 20 amino acids. We have 64 possible letter combinations to code for the 20 amino acids. We have more codes, more codons, than we have amino acids. That means that some amino acids will be coded by more than one 3-letter sequence. Leucine, for example, is coded by UUA, UUG, CUU, CUC, CUA, and CUG. Notice also that there is one codon that to the cell represents the beginning of the polypeptide or the protein. The AUG codon is the start sequence. It actually codes for an amino acid called methionine. In prokaryotic cells, this amino acid is modified to formyl methionine. So AUG is always the first codon in a messenger RNA that will be translated. Also notice there are 3 stop codons, UAA, UAG, and UGA. During translation, when the cell arrives at this codon, that is the signal that the cell has finished synthesizing the polypeptide and translation stops. Finally, notice that the orientation of the codons is the same as the orientation of translation. Messenger RNA is translated 5' to 3'. So the codons are written in 5' to 3' direction. This first U in UUU is in the 5' position. The last U is in the 3' position. The question is, how does the cell correlate a codon sequence to an amino acid sequence? The key is in another RNA molecule called the transfer RNA or tRNA. The tRNA is a clover leaf-shaped molecule made of nucleotides. It is, after all, an RNA molecule. At one end of the molecule, there is always a three-codon, three-nucleotide sequence called the anticodon, which will match with one of the codons in the messenger RNA. At the other end of the molecule, the amino acid that corresponds to the codon will be attached to the tRNA. So the tRNA with an anticodon UCA that corresponds to the codon AGU will have attached a serine nucleotide at its other end. When you look at the genetic code, one of the codons for serine is AGU. AGU. The tRNA with the codon UCA can base pair with AGU. The tRNA with the anticodon UCA will always have a serine attached at its other end, thus matching the codon with its corresponding amino acid. Here is another example. The codon in the messenger RNA for tyrosine is UAC. The corresponding anticodon that will complement UAC in the tRNA that carries tyrosine would be AUG. When you look at the genetic code, UAC is the codon for tyrosine. The tRNA that carries the anticodon will have tyrosine attached at its other end. During translation, there will be a multitude of tRNAs carrying all possible amino acids, which will complement the codons in the messenger RNA, and the polypeptide sequence will be formed. Again, remember, tRNA is an RNA molecule made of nucleotides, A, C's, G's, and U's. This is the anticodon region of the tRNA molecule. On the other hand, ribosomal RNA is made of a combination of RNA molecules and polypeptides. So it's a mixture of RNA and amino acids that creates the ribosomes. The ribosomes will be the location, the place where translation will occur. Remember that eukaryotic ribosomes are made of two subunits, a large 60S subunit and a smaller 80S subunit. Once both subunits are assembled, an 80S ribosome is formed. Prokaryotic ribosomes are smaller. They have a 50S subunit and a 30S subunit. Once both subunits are put together, a 70S ribosome forms. S stands for Sedberg units, and these are the units used to measure the sedimentation rate of the ribosome, which roughly depends on size, molecular weight, and shape. You should now be able to answer the following questions. Determine the location in the cell where prokaryotic transcription and translation take place. Determine the location of the cell where eukaryotic transcription and translation take place. Could transcription and translation occur simultaneously in prokaryotes? What about eukaryotes? Given a sequence of DNA, you should be able to transcribe and translate, given a genetic code, the sequence. So for example, given this DNA sequence with these codons, you should be able to choose one of the appropriate DNA strand to transcribe, making a messenger RNA, and then use that messenger RNA with a genetic code and translate the messenger RNA.